Hello, my name is Courtney Dziewior, and today I will be presenting on you on my research regarding the synthesis of stimuli-responsive programmable polymers through the ring opening metathesis-cross metathesis reaction. This research has been done with the collaboration of Ken Shimizu's lab at the University of South Carolina, as well as Ping Li, who previously worked in his lab, and my amazing mentor and professor, Dr. Sherry Strickland. Additionally, the research would not have been possible without the generous funding by the South Carolina INBRE program, as well as the NSF and South Carolina EPSCoR IDEA program. I'm very grateful to both of these entities. First off, what is a stimuli-responsive polymer? Well, a stimuli-responsive polymer is, as the name suggests, something that responds to a stimulus. So the stimulus can be any sort of stimulus you can imagine. The most common, though, are light, temperature, pH, solvent, and magnetic field. So whenever a polymer recognizes the stimulus, it'll respond to it. And so this is why these are known to have adaptable recognition properties. What's unique about the properties in the polymers I'm studying is that they are also programmable polymers. So a stimuli-responsive programmable polymer has a memory and reversibility capability. And so it can recognize a stimulus and change, but also recognize when the stimulus is gone and memorize its response. And so these types of polymers have a very wide variety of application potential. And so one such application would be controlled drug delivery. The polymers can be programmed on where to deliver the drugs and when to release it. Additionally, they're also used for biosensors, magnet behavior, artificial muscles, and for protective coatings. So they have a wide variety of applications. So the methods for my research has involved a series of reactions to identify and synthesize the functional monomers in order to then polymerize them further down the road. And so what you see here is the first reaction scheme that I studied in regards to the maleic acid olefin. And so the first reaction that you see that produces monomer 1B is a condensation reaction. And so that reaction has been very successful in the past. My previous work has been able to identify and characterize the monomer 1B. What this project focused on is on synthesizing monomer 2B and then later on monomer 3. And so monomer 2B is formed through a ring opening metathesis-cross metathesis reaction with the olefin maleic acid. And what happens is the double bond at the bottom of monomer 1B will break. It'll cross with the maleic acid and form monomer 2B with the carboxylic acids at the end of it. And then once monomer 2B is synthesized, it is a carboxylic acid, and those are hard to isolate and purify. But once it can then be esterified for our polymerization, which would result in monomer 3. And so the one thing with this reaction, and I will report on the data in a moment, is that it is not strained. The original monomer is an unstrained cyclic alkene. And for the polymer, we are interested in a strained cyclic alkene. And so that's why we looked at this reaction scheme as well. This reaction scheme is virtually the same. The difference is that it possesses a bridge on the anhydride that you see at the very bottom. And so that means that whenever the ROM-CM reaction occurs with maleic acid, that bridge will fold down, the double bond will break and cross with the maleic acid. And so you just have that slight structure difference. But the rest of it's the same. It will produce a carboxylic acid for monomer 2C, and then it will need to be esterified for future polymerization efforts, which would be monomer 3C. So the first work that I'll be reporting on is with the unstrained maleic acid reaction scheme. And so just a reminder, that's what you see here on the right, and we're trying to identify monomer 2B. So whenever I perform this reaction, I generally follow all of my reactions using TLC, also known as thin layer chromatography. And so what TLC does is it identifies what's present in the reaction mixture at the time that it is spotted on the TLC plate. And usually the way this is done is by comparing with other things that you have already identified in the reaction mixture. And there are also different ways of visualizing it. The most common is what you see on the left, where it has a green light to it. That is UV. The middle one is using a potassium permanganate stain. And then the right one is a bromocresol green stain, the one that is a blue plate. And so the UV light is the most indicative of how many spots are present in the reaction mixture. Each spot represents a different compound, and according to that one, there's 3 different things present in my reaction mixture at the time that I took the TLC sample. And the middle one, it says that those 3 were all alkenes. And then the right one is the bromocresol green stain and was the most useful for this. And so I know that in these 3 spots, the top spot is monomer 1B. I was able to identify that by using samples of monomer 1B. And the bottom spot is maleic acid, but the middle one is unknown. The fact that the middle spot is large and the maleic acid is small means that the middle one is likely a product from this reaction, and it could be a side product or it could be our monomer 2B. And the fact that it was reactive in this stain means that it's a carboxylic acid, as only carboxylic acids react and turn yellow in bromocresol green. So this gave us pretty much every indication that this was our monomer 2B, which was really good news. And so since this was with the unstrained version, which we can't use in future polymerization efforts, I decided to go back to the highly strained reaction scheme and investigate that one because the unstrained had such good results. So with the highly strained reaction scheme, I did conduct the reaction in where I got the monomer 2, where it had the high strain with the bridge. And then the issue with this is it's not easy to analyze using TLC because of the high strain with the carboxylic acid. So I first had to isolate it using a reverse phase column chromatography, which is what you see on the left. And so the reverse phase column chromatography is used for things that are more difficult to isolate, such as carboxylic acids. And so the results of this are still inconclusive, as the products were isolated and one was found to just be a side product of the salts that were present when doing the column chromatography. But the second product that was isolated is still being analyzed at University of South Carolina in Columbia. And so you see on the right, these are the TLC analysis of the fractions that were taken from my column, and I isolated the spots, but as you can see, it's very streaky and hard to visualize, and that is because they're carboxylic acids, and most of the time, they're very difficult to see. So the results are still waiting on whether I was able to produce the desired monomer 2, and then be able to esterify it to monomer 3. But I made significant headway. So in conclusion of my project, I was able to see that monomer 2B with the low strain was easily synthesized. And so the monomer 2C with the high strain is still waiting on further details, but hopefully it was successful, and if so, I will be able to continue with future work on esterifying it and characterizing it, and then ultimately polymerizing it into our stimuli-responsive programmable polymer, and be able to do property studies and see if it does possess the properties we want it to. So thank you for listening to my presentation. I hope you found it interesting, and I hope you enjoy the rest of the conference.